Okay, thank you all to being here. I'm going to talk about of our project that is the Imagine Flow Cytobot and face data for phytoplankton communities carried by Christian, Amir, Lindsay, Jose, and myself with our project mentor, Cara Wilson. Ah, okay. Talking about a little bit about phytoplankton that are we know that are single cell plant-like microbes that use sunlight to build organic matter, drawing down CO2 and releasing oxygen to our planet Earth. Uh, their striking diversity in size, shape, and life cycles let them thrive in nearly any sunlit ocean from the polar seas to tropical reefs. So in this project, we combine two optical tools for study the phytoplankton community structure using the IFCB, Imaging Flow Cytobot, acquire optical and image underwater data of phytoplankton, then transmitted to the Martha's Vineyard Coastal Observatory shore in real time, and also the NASA Space Mission hyperspectral data. By pairing the space and time coverage of phase and the EFCB imaging data at the fixed point, we aim to bridge the scale gap between what the ocean looks like from space and who is actually there in the water, moving toward more robust composition-aware monitoring of phytoplankton diversity and its ecological significance. Talking about the site location, there's a tower located a mile of the south shore of Martha's Vineyard and the meteorological mast on the south beach of Martha's Vineyard. The instrumentation at these sites provide real-time and archived coastal oceanographic and meteorological data. So I pass the voice to my Christian.
Oh, I guess my webcam's pointing away from me, but yeah. So I did one of the main data pipelines. So essentially, I loaded the data set we had, and we lost our original data set. So we had to do some quick changing to make this work yesterday. Um, but we took all the cell abundances from the IFCB data, looked at the dates essentially that they were recorded at, as well as obviously the location. Then we searched all the paste data for that, and because if you use nearest, for example, an X-array to search for the data, some days are going to be cloudy, so you're not going to find the exact pinpoint measurement for that day. So you'll have to I took like a 4 kilometer radius around the point, as well as a 1 day basically temporal constraint, and found a lot more matches that way. Otherwise, you could only match approximately like 200 of the data points to satellite data versus I think we ended up with a lot more. I think it was like 400 or something like that after the matching. Nice person that. I don't have much to talk on this. I don't know if Amir is here.
Yeah,
Um,
I'm here.
okay, yeah.
Yeah, so sorry to jump in. For this part, we did actually the data set that we had, like the CSV file, includes like lots of species, like hundreds of types of species, which makes it very difficult to analyze. So that in this step, we just categorize them. Like we try to do like to categorize them based on the like broader phytoplankton types, like diatoms, dinoflagellates, and so on. So this is the schema that we used to do that. And then we also categorize them based on like being micro, nano, or pico phytoplankton, or like another category that we use is silica versus non-silica, knowing that like diatoms are mostly like silica, and the other types are non-silica phytoplankton. So next slide, please. Yeah, so we're actually so this is the first analysis that we did. We tried to relate the optical water type to the abundance of each category of phytoplankton in the water. This slide, the top graph shows the abundance of each group of phytoplankton in each optical water class. Uh, so for example, we see higher, obviously lower optical water class means probably more clear water versus the higher optical water class which means the turbid water. And we see some types of phytoplankton groups has different abundance in these different water classes. For example, we see for example, we see lower dinoflagellates in turbid waters versus the green algae, which are more prominent, more abundant in turbid waters. This graph basically shows how spectral reflectance change with the phytoplankton composition. And also in the lower side, you can see how AVW parameter change with the phytoplankton groups. Yeah, back to Carol.
Yeah, applying what we learned, here we made the graphics for the optical water class and the average visible wavelength from the side point, obtaining that the optical water class predominance was the class number 10, and the visible wavelength predominance was from 510 to 520. And Amir, I think that you're next. Yep.
Yeah, this is the results of another analysis that we did. So we try to go further than water phytoplankton types to more toward the size of the phytoplankton. So we categorize them based on being micro, nano, and pico, and did this analysis to show the abundant the water type of the water type for each of these categories. So we saw that obviously we had a good match between the water class and ABW, which was expected. So more the higher number for water class matched to the reddish greenish, yellowish, and reddish water as we expected. And then we also saw that microphytoplanktons tends to be more in greenish water compared to the blue water. What we didn't expect it here was to lower microplankton, almost any plankton type in yellowish water compared to greenish water. Something that we probably need to dig into it a little bit more later. Next slide, please.
And here we did the exercise of the AVW from the range from 4990 to 550 nanometers to see the changes of the wavelength on what the graphics from the previous slide show.
You're
I
okay.
think okay. I think this is Jose.
Yeah, we here basically showed the chlorophyll derived for PACE, and we compare with the nanoplankton METS abundance of the data set. We see basically this temporal trend And the next, please, Carol. Amir.
I think this is not mine actually.
Okay, but
Okay,
yeah, it's
Sorry.
basically Christian, I think so.
Yeah, so I plotted this basically to just show the correlation between the satellite abundance and the IFCB abundance and the nanoplankton mix. Not exactly sure, like I'm not an expert on phytoplankton, but basically my takeaway from this is that a spike in the abundance of the cells measured in the IFs and the paste data doesn't necessarily mean a spike in the carbon biomass, right, of the IFCB data.
And when we look the correlation matrix here, basically you in the first row, chlorophyll and VGC carbon phytoplankton, you look this correlation with the phytoplankton community from the EFCB. And you look the maximum correlation related with some groups that we can Amir explain in the next slides.
So again, Christian, I think it's yours. Christian.
Yeah, so this is also another graph trying to show the relationship between the chlorophyll and this is the one from I believe level 2, as well as it takes the group of diatoms generated by Amir and tries to just classify that abundance of the counts versus the chlorophyll data. And then this is also just an abundance versus the which one is that? The carbon POC?
Yeah, carbon.
POC, yeah. The POC value from PACE.
So with that, our challenges were that we have to rush into a new project because we lost our original data and changed the direction of the project using the imaging phytoplankton data from the optical bot. Another challenge was the categorization of phytoplankton community based of some morphological species, traits like size or use of silicate or other traits. The future directions that we can take from this project is to explore the potential application of base data in the taxonomic identification of the phytoplankton community based on this imaging and use the base data in our research questions. And we like to thank Ellie and Ryan, our lead organizers and project mentors, for the amazing information and mentoring throughout the HackWeek project process, to the incredible science lecturers for their values and interesting topics, and to all the project mentors for their time and advice, and also to thank the sponsors. And that's all. Thank you.